share the screen with the PowerPoint and the whiteboard. So if you do, please send me a reply in the chat room to let me know. Okay, so I can see 3 replies only, and I just assume everything is working. So before we start to talk about the other types of biological sample for the poison analysis, so let us have a brief summary for what we have been talking about last week. So last week we have been finished to talk about the drug analysis, okay? So for the drug analysis, we start with the visual examination. So in view of the visual examination, actually we are checking with the drug itself for the grouping. Okay, so we try to group the drugs, okay, as the individual sample. So in another way around, we try to do a visual examination on the plastic bag which contain the drug sample for the comparison purpose in order to check back for the origin. So let's say the drug manufacturer or the drug seller. So after we are finished with the visual examination, then we try to make use of the screening test to further narrow down our number of possibility. So for the screening test, we have 2 different kinds of screening tests to be applied. The first one that is related to the color test. So you have the drug sample added with the reagents, then color will be given. So we have 1 preliminary color test, 5 primary color tests, and quite a number of secondary color tests. So other than that, we can apply the microcrystalline test, okay, so for the screening purpose. So basically, we have the drug sample added with some reagent, then crystal will be formed, okay? Then we observe the crystal under the dark field microscope, okay? So based on the size and shape as well as the color, then we can be able to identify what kind of drug it could be. So after the screening test, then we try to go through some sample pre-treatment. So for the sample pre-treatment, that will be quite straightforward and simple, okay? Because of the drug sample, the matrix that is quite not that severe. So therefore, it will be simple that we can go through the complete the solution. So just try to dissolve the whole drug sample as the sample solution. So if you just worry a little bit on the matrix, so you could perform a simple extraction. So try to get rid of some of the matrix, okay, so that the sample solution will be much cleaner. Then matrix effect will not be that significant. So after the sample solution is being prepared, then we can go forward for the confirmatory test, and even we can go for the retest, okay, for a double confirmation. So for those kinds of confirmatory tests, we have to achieve the identification to know what kind of drug it is. So at the same time, we have to perform the quantification to know about for the respective amounts, okay? Because for the drug analysis or the drug sample, the amount it is really important. So for those kinds of confirmatory tests, okay, there are certain kinds of instrumental analysis. So we can make use of the chromatographic techniques, okay, so to help us to deal with the confirmatory test. So it could be a TLC or it could be a GC. So other than that, we can make use of the spectroscopy to deal with the confirmation, okay? So it could be a UV visible, or it could be the IR, or even for the Fourier transform infrared, as well as the Raman spectroscopy. So we still, okay, have the techniques, okay, so on the mass spec, okay, so mass spectrometry, so as the confirmatory test. So for the mass spec, it could be just simply a biological mass spec. Okay, so that will be a ESI quadruple or the multi-TOF, or it could be a tandem mass spec. Okay, so the MSMS, or even we would like to make use of the hyphenated technique. Okay, so such as the GCMS. So these are the instrumentation that we can use, okay, for the confirmation. So basically, with the use of the instrumental analysis for each of the techniques, we have different kinds of parameters, okay, so for the identification and also for the quantification. So at the same time, one thing for you to further notice that is whether the analysis it is destructive or non-destructive in nature. Because for the first confirmatory test, ideally it should be non-destructive, so that we can retain as much as sample as we can, so that we can have sufficient sample to run for a retest, okay, for a double confirmation. So for the TLC, the parameter that we use for the identification will be the retention factor. So that is the distance of travel, okay, so on the thin layer substrate. And for the identification, that will be tentative only, okay, because we just separate the substance based on the polarity only, okay? So it may have similar kinds of polarity for different compounds, and the distance of travel will be quite similar, okay? So for the GC, we base on the retention time, okay? So the time that you look out from the GC column, and the identification will also be tentative, because for the GC column, that will be the polarity only, okay? So compound substance with similar polarity, they may have a similar retention time. For the UV visible, we identify through the absorption maximum or the absorption minimum, okay? But the identification still be tentative only, okay? Because different kinds of compound, if they share similar kinds of structure, actually they will show up as similar absorption peaks, okay? So for the IR, Fourier transform IR and the Raman, okay, we identify by the mid-infrared spectrum. So for the mid-infrared spectrum, as far as we know, it is a fingerprint region. So that different compound, okay, different substance, they will show up different kinds of peaks, okay, that is regarding to their functional group. So that the mid-infrared spectrum that is unique, that the identification will be confirmative. So for the mass spec, okay, so no matter that is the original single mass spec or tandem mass spec, we can identify the compound by the respective mass to charge ratio. But to be specific, actually we can make use of the whole mass spectrum, okay, so for the identification purpose. Because apart from the parent iron, okay, we still could be finding some fragmented iron, okay, so those kind of fragmentation of the original compound, that will also be specific and unique, okay, to that particular kind of compound. So for the GC mass, we are making use of the retention time together with the mass to charge ratio, the mass spectrum together to identify what it is. So altogether, if right now we are having the mass to charge ratio, having the mass spectrum, that identification will be confirmative. So in view of the quantification, so TLC, we cannot do anything for that. And for the GC, we can make use of the pit area for the identification. So UV visible, as you know, that will be the Beer's law. So that will be the absorbance for the quantification. Original infrared, we cannot do any quantification because that is semi-quantitative only. For the Fourier transform infrared, based on the transmission or the absorbance of a particular infrared absorption peak, then you could be able to perform the quantification. For the Raman, that is the light scattering. So therefore, that will be depending on the scatter light intensity. So for the mass spec, that will be the relative abundance. Okay, so that is the y-axis of the mass spectrum. And for the GCMS, that will be existed as a pit area. Okay, So these are the parameters that we can use, okay, so for the quantification purpose. But all in all, okay, in order to achieve the quantification, basically we need a calibration curve, okay? So if we don't have any calibration curve, then actually we do not have any quantitative information. So at last, okay, so TLC, that is non-destructive in nature. And then for GC, depending on what kinds of detector that you are using. For example, like if you are using the FID, then that will be destructive. If you are now using the TCD, then that will be non-destructive in nature. So all the spectroscopic techniques, they are non-destructive in nature so that you can get the sample back, okay, for the retest. For the mass spec, altogether, they are destructive in nature, okay? So whatever you send in, so they will be just strongly ionized, fragmented, then actually everything will be burned, okay? So that is all the stuff that we have been mentioned about in the drug analysis. Okay, so any questions or problem at this moment before we move on to the next topic? Okay, so seems no question at this moment. So last time we have started to talk about a new topic on the forensic toxicology. So for the forensic toxicology, most important thing that is related to the poison. So something that is toxic, and when a person is subjected with a poison, so normally negative effect will be given. Okay, so that is the opposite of a drug. Okay, so drug by default, that is something that will give you some positive effects. But for the poison, okay, so by default, okay, it will give you something negative. So trying to hurt you, try to cause you injury, or somehow try to kill you. So for the poison, we could be compared by the threshold, okay? So that will be the minimum amounts that start to give you the effects, or even it could be compared with the LD50, so that is the Levin dose 50. So in view of the two different kinds of amounts, okay, if a person is subjected to a poison at the threshold level, somehow for a long time, so that will be a long-term poisoning cases. And for the LD50, the dosage that is really high already. So somehow within a short term, okay, the effect will come out somehow immediately. So somehow with the poison, basically the amount, the dose, it is really important, okay? If it is low enough, then it may be somehow a long-term effect. If it is high enough, maybe the effect will come somehow immediately. Or somehow if something by default it is non-toxic, if right now it is subjected to a very large amount, it will still become something toxic, okay, like water. So therefore, for the poison, apart from the identification, quantification that is still necessary. So for the poison analysis, basically, we are suffer from a lot of difficulty as far as you know, okay, the concentration it is at the ppb level. We have to extract out those kind of poison from the biological sample. Therefore, for the whole analysis, it is quite tedious, okay? So somehow rigorous sample pre-treatment and also certain kind of sensitive technique to do so. But before we really go for the analysis, so the next thing that we have been talking about that is related to the sample, okay, that to be taken, okay, for the poison analysis. Because right now the poison itself, it is not a poison alone. The poison has been subjected to someone's body already. So therefore, we have to take out the poison from someone's body, okay? So in our body, we have different kinds of sample to be chosen, but we have some parameter, okay, to guide us, okay? So what kind of sample will be somehow the most appropriate? So for the parameter that we need to consider, that will be related to the objective, okay, the relevancy, and also the type of poison to be analyzed, at the last, okay, that will be the availability as well. Okay, so last time we have started to talk about two of such kinds of sample already. So the first one would be the blood sample, okay? And the second one that we have been talking about, that would be the urine sample. So for the blood sample, as far as you know, okay, so when someone that is subjected with the poison somehow orally or somehow through the muscle injection, okay, so actually the poison, if you get it orally, so you will just go through the stomach, after 2 to 3 hours will be digested, going through the intestine to be absorbed, to be circulated through the blood system. So if it is injected, then through the muscle, then will still will be absorbed by the blood, so still will be circulated. So therefore, the poison will be appear in the blood sample somehow 2 to 3 hours later. So therefore, the poisoning issue will be quite of a short term, okay, because they will be within hours. So since, okay, all the poison will be absorbed through the blood system, so therefore, apart from identifying what are they, the quantity will also be quite important, that will tell you the dosage, okay, of the original subjection. For the types of poison to be present, at the very early beginning, that will be the poison itself, okay, but later on, the respective metabolites will come over, okay, because the blood will transfer the poison to the liver for the detoxification, so that with certain kinds of metabolism, the metabolites will come. For the blood sample that will be available for a live person, okay, if someone is died for some time, then actually you cannot take out any blood anymore. So that is the situation for the blood sample. For the urine sample, that will be also by dealing with the short-term poisoning cases. So mainly because, okay, once the blood has been transferred to the liver, those kind of poison will be going through the metabolism, so that the metabolites, so that will be excreted out from the urine, okay? So therefore, that will be seeing those kind of metabolites around 6 to 8 hours. So after a person is subjected with the poison, so therefore it will still be hours, okay? So therefore, there will be short-term poisoning. But for the urine, okay, you can achieve identification only because the quantity that is quite meaningless. So different person, they will have different metabolic rates and also excretion rates. So therefore, the amounts appear in the urine, okay, you cannot tell you about the dosage because there are some individual factor that is individual dependent. In the urine, you can only find the metabolites only because only metabolites can be excreted out. And for the urine, that will be available for the alive person. So just ask him or her, okay, to go to the washroom and then just get in with the plastic vial so you can collect the urine readily. So these are the two kinds of sample that we have been talking about last time. So for today, we are going to talk about the rest of the sample. For example, like the hair, saliva, different kinds of organs at the same time with the insect. So after we finish to talk about different kinds of sample, then we will go ahead to talk about the analytical protocol. So right at this point, any questions or problem before we go forward? Okay, so thanks for the reply. Then I assume no question at this moment. Then we start to go forward with another kind of sample. So that is the hair. So for the hair sample, that will be quite unique, that is capable for recording long-term poisoning cases. Okay, mainly because, okay, so you can be imagine that now a person is subjected with the poison, so that the poison will be carried around, okay, within our body through the blood system. So when the blood, okay, has been going to our head, okay, so somehow at the beginning of our hair, so that is the follicle, okay. So the follicle, that is the region that the hair will absorb the nutrient from the blood, so that the hair can be grown, okay. But at the same time, not only absorbing with the nutrient, okay, But for those kinds of poison and the respective metabolites, so they will also be accumulated inside the follicle. Okay. So therefore, inside the follicle, if there are some poison appear in the blood, so they will be accumulated there. So those kind of metabolites and the poison will be go along, okay, with the hair. So what I mean is those kind of poison and the respective metabolite, they are being trapped, okay, so inside the hair. So since, okay, the hair will be growing for 1 to 1.5 cm per month. So therefore, the hair will be keep growing. And then for each of the length within the hair will be representing 1 month and then the other month. So therefore, for each of the length, okay, they were telling you that whether at that period, okay, is it the person has been poisoned or not, okay? So because what I mean is once, okay, there are some poison and the metabolites in the blood, they will be accumulated there. If they are there, then they will be somehow grow along the hair, and then you will be seeing them within, okay, a certain length, okay, in the hair sample. So therefore, right here, what you can be imagine is there will be a long-term poisoning cases. At the same time, there will be the poison and the metabolites together. So you can see, okay, if now we have a hair sample, so that is the follicle, that will be the starting point 0. So if you have a long hair, okay, so that is a long hair, okay. So for the first 1 cm, okay, that may be somehow that is the March, okay, so this month, and then another 1 cm, so that will be the February, so that is the last month, okay. So another 1 cm January, another 1 cm December, another 1 cm November, another 1 cm October, so another 1 cm that will be September, and then to be August, okay. So actually what you can see is with a sufficient length of the hair, we can trace back to somehow like months, okay? Or even for the lady, okay, it could be somehow for a years, okay? Actually, that will be providing the information for the track record, okay, for the long-term poisoning, okay? So maybe the guy has been poisoned in November, then the next dose it is subjected in January and then March, okay, it also be having some poison there. So actually, we can be able to tell, okay, so what kinds of poisoning things are there, and then with the time period, okay. So therefore, by using that, that will be quite important, because for the blood sample, there will be hours. For the urine sample, there will be hours or even just last for 2 weeks. But right now, with the hair sample, that will be months or even for years. So actually, that will be a very unique kind of sample that we are checking for other kinds of poisoning cases, long-term poisoning. So for the relevancy, actually there will be no doubts that you could be able to identify what kinds of poison are there. So based on the poison or the respective metabolites accumulated inside the hair. But for the quantity, that will be meaningless, okay? Because for those kind of poison and the metabolites, while they are accumulated in the follicle, actually that kinds of deposition will also be dependent of the individual. So different person, they will have different deposition rates, okay? So therefore, some people may have more, some people will have less. So therefore, what we can do is we can only identify there is something, but for the quantity, it will give you something meaningless. So you don't know what is the dosage right at that moment. So therefore, for the relevancy of the hair sample, that will be identification only. So for the availability, that will be quite straightforward. So if you are a alive person, you have hair, then you can cut the hair out so that hair sample will be available. So for the person has been died, okay, for a short time, okay, so actually the hair will still be there. So therefore, for a dead person, you still could be able to obtain the hair sample, but not trying to foresee that the guy has been died for a long time. So after a long time, the hair will also be decomposed. So only within a short time, okay, after a person is died, okay, you still could be able to take the hair sample from there. So in order to take the hair sample, that will be quite easy and quite straightforward. So simply you just take the hair and then you make use of the scissors to cut, okay, as close as the scalp, okay? Because what you need is you need the time 0. Okay, if you just randomly cut out some part of the hair, then actually you don't have any reference point, okay, to be the 0. So we need the 0 point, and then the first 1 cm will be this month, another 1 cm will be the last month, another 1 cm will be the last month. Okay, so actually there will be something that we need, okay, the point 0. So right here, you may have a question that whether the poison and the metabolites, while they are stored inside the hair, are they stable? So will it be leached out? Okay, if some people make use of the hair dye, or you wash your hair. So actually, are they stable there? Will they be leached out? Actually, those kind of poison and the metabolites, they are stored somehow at the core, okay, at the core center of the hair. So no matter how you do it, okay, you can only remove something at the outside. So something at the inside will be just quite stable and then will be last forever. So no matter a normal person how they deal with their hair, so those kind of poison or the metabolites will not be washed out. So they will be there if you are really being poisoned. So here just shows you some detection time, okay, when we try to compare the blood sample, urine sample, and the hair sample. So as far as I already mentioned, so blood sample, we will start to see it after 2 to 3 hours. For the urine sample, we start to see it after 6 to 8 hours. For the hair sample, that will be months, okay, or for some lady, okay, that will be last for a year, okay. So once you have hair, then actually we can take the hair sample to check whether that is a long-term poisoning cases or not. So right now you may have another question. So some people in the world, okay, they don't have hair, okay, then how we can be checked with the long-term poisoning cases, okay? The answer is we could make use of another kind of sample. So that is our fingernail. Okay, so we can make use of our fingernail, okay, to replace the hair sample to check for the long-term poisoning cases. So basically the logic will be quite similar. So imagine that is our finger, okay, so our fingernail. So at the beginning of the fingernail, actually we are full of the blood vessel. So the fingernail have to be absorb the nutrient from the blood in order to grow up, okay. So actually, if we have some poison there, they will also be deposit accumulated there. So they will go alongside with the fingernail and then going up and going up and going up, okay. So therefore, along, okay, with the fingernail, okay, so actually you can see, okay, the poison, okay, with the sufficient time frame there, okay. But right here, it will be applied to some people, they do not have hair, then you take the fingernail, okay. Because basically, if you try to take the hair, that will be quite straightforward. You just cut it, and then the person, okay, so will not feel any pain there. But if you try to take the fingernail out, it will be quite painful, okay. So if you try to making use of some terms, okay, to say that hair sample that is non-invasive, and then for the fingernail that is invasive, okay. So therefore, normally if a person has hair, then we take the hair sample. So other than that, okay, if a person doesn't have hair, then you have to try, okay, with the fingernail, okay, from him or from her. So another kind of sample that I'm going to talk about that is related to the oral fluid. So for the oral fluid, in a layman term, that is the saliva. So for the saliva sample, that will be gaining a quite important nowadays. Because for the saliva, it will be another kind of blood sample, okay? So it is not saying that we try to take out the saliva because when someone has been poisoned orally, then some poison residue that is left behind in the mouth, okay, so that we take the saliva. Actually is, if you know about how human, okay, so generate saliva, actually you will know, okay, saliva that is a filtrate, okay, so of the blood. We just take it quite simple sense is, so we have the blood and then we go through the saliva gland, okay, so with certain kind of filtration, then that will become saliva, okay. So actually, if you think it like this way is blood after filtration, then it will become saliva. So actually blood and saliva, they are equivalent, okay. So therefore, not taking the blood sample, we can make use of the oral fluid, making use of the saliva, because they are the same. Okay. So therefore, for the saliva, simply it is the same as blood. Okay. So short-term poisoning cases, the dosage can be reviewed, and then whatever kind of things in the blood, they will be appear in the saliva. Taking the saliva will be available for the alive person. Actually, for the blood and the saliva, they are the same. Okay. So how we can take the saliva from the victim or take from somebody? So basically, we can make use of the cotton blood. Okay, so open your mouth and then you just make some scrub. Okay, then you can take the saliva from a person. But making use of the cotton blood, okay, in order to scrub the saliva, the amount may not be that sufficient. So therefore, in order to collect large amounts of saliva, so we are going to make use of some plastic vial, okay? So you have to ask a person to open their mouth wide and then to let the saliva to leach out passively, okay? So we don't want to collect those kind of bubble, okay? If you actively just take out the saliva, okay? So just open your mouth wide and then let the saliva to be leached out passively, okay? Then you can collect it. Then for a large amount for the poison analysis. So for the oral fluid, as far as I mentioned, that is just the same as blood, okay? In view of the detection time, but it will have a certain kinds of a little bit delay, okay? Because for the blood sample, that will be 2 to 3 hours. For the urine sample, 6 to 8 hours. For the oral fluid, that will be something in between, okay? Because what you can be imagine is, so now you take it orally, digest through the stomach, and then absorb it by the intestine, and then circulate around the body. So that is the blood sample 2 to 3 hours. So once the blood, okay, going through the saliva gland and then becomes saliva, actually that will be more than 2 to 3 hours. Because the blood, okay, appear with the poison. Later on, they transfer through the saliva gland, go through the filtration to become saliva. That may be more than 2 to 3 hours, but it will be less, okay, than the 6 to 8 hours. Because right now, it is just a filtration. It doesn't go through the liver with the detoxification, or even didn't go through the kidney, okay, so for the excretion, that is the things, okay, happening in the urine, okay. So therefore, if you are now checking with the detection time for the saliva, actually that will be in between, okay, the blood sample and the urine sample. So right now, what you have been seeing is from the basic biological sample, or for the primary specimen, so blood sample, urine sample, hair sample, or even from the oral fluid. So most of them, okay, they are available for an alive person, okay? So if now, okay, a person is died, okay, so somehow has been died for some time, okay, we cannot take the blood, okay, because the blood has been dry. We cannot take the urine, okay, because you cannot ask a dead person to go to washroom. And then you cannot take any oral fluid there because it is already dry as well. For the hair, there will be long-term poisoning cases, okay. At the same time, if he or she has been died for some time, no hair there, then how, okay, we can be able to conduct the poison analysis? So with a dead body. So with a dead body, so basically we can take out something, okay, so inside the body, okay? Because for a alive person, you cannot just cut the body and then take the organ. But right now, with a dead person, then you could be able to cut the dead body to take out the organs, okay, for the poison analysis. So basically, to take out the organ that will be available for a dead person, okay, and then to cut the dead body, basically we make a Y-shaped cutting. So we have a Y-shaped cutting so that you can be open the body and then seeing those kinds of organs. So right now the question would be, what kinds of organ, okay, should we be taken? We cannot be so greedy that to take all the organ, okay, out, okay, for the poison analysis, okay. If you would like to take all the organ out for the poison analysis, actually you make yourself in a trouble. because after you conduct the analysis, you have to put the organ back to the dead body. Okay, so it will be just like a puzzle. Okay, so you have to fill up all the organ back to the body in a correct position. So therefore, we have to quite making sure that we have to take the appropriate organ. Okay, so I will show you each of the organ for the respective importance somehow in details. But in general, okay, so for the organ, we can't tell, okay, any objective there because it could be within a short time or it has been accumulated deposit for a long time. Then we don't know which one is which. Okay, so therefore we cannot tell about the objective. Okay, and for the relevancy, there will be identification only. Because inside the organ, basically we are saying with the accumulation rate or the deposition rate. And even we need to count with the excretion rates, how much of the poison has been excreted out. Okay, so therefore inside an organ, how much are there? That will be affecting by a lot of individual factor. Okay, so therefore we cannot know, okay, the dosage at the very early beginning. And whatever kinds of things appear in the body, so no matter that is the poison or the respective metabolites, so they may be accumulated, they may be deposited, and then they will be appear, so that they will be the poison together with the metabolites inside the organ there. So right now, let's go to talk about each of the organs somehow in a little bit details. So one kind of common optos sample that is our stomach. Okay, so why we want to take out the stomach? So basically, we want to check with the stomach content. So what kinds of stuff has been left behind in the stomach? Okay, because what we can be imagine is if someone has been poisoned orally. Okay, so imagine that you take the things through your mouth orally. The first place that we enter the body would be the stomach. Okay, so if the person has been poisoned within a short time and then the effect is quite strong, he or she maybe die, okay, within, let's say, 2 hours. Okay, so actually, if you still recall what I'm saying is the stomach, okay, need to take 2 to 3 hours to digest the stuff before it open to get those kind of stuff to the intestine for the absorption. So therefore, if the guy has been being killed, okay, being poisoned within 2 hours, actually you can find those kinds of poisoning things, okay, in the stomach. You may be finding some undigested pill, undigested liquid, or undigested poison. Actually, that will be a quite useful information from the stomach. So apart from that, from the stomach, actually we can be used, okay, to estimate the time of death. But basically, that will be less than 2 hours or more than 2 hours. Okay, because what you can be imagine is if someone is died, okay, then the digestive system will be stopped. Okay, so if you can find something in the stomach, that means, okay, within 2 hours. If there is nothing in the stomach, then that will be more than 2 hours. Okay, so other than that, okay, if now you cannot find anything in the stomach, actually, after the digestion, there may be some cell, okay, of the fruit plant, okay, has been left behind as certain kinds of residue. So actually, from those kinds of characteristic cell type of the fruit plants, you could be able to tell what kinds of food has been subjected to that person. Okay, for example, like we have some. Oops. Okay, so just wait for a moment. What happened to the PowerPoint? Okay, so just wait for a while. So let me share the PowerPoint again. I cannot do anything on that. Okay, so maybe let me reopen the PowerPoint. So just wait for a while. So please wait. Okay, Kosen, can you see my shared screen with the PowerPoint? Okay, so there's something happened that I cannot make any animation from the PowerPoint. So actually, I don't know what happened. Maybe I share the screen here. Okay, so you can see the picture. So on the right hand side, okay. So actually here showing you the characteristic cell type, okay, of certain kinds of fruit plants, okay. Actually, that is something that as a residue left behind in the stomach, okay. So which, okay, we can be used to identify what kinds of stuff has been taken by the victim, okay. So therefore, the top left-hand corner, so that is the characteristic cell type of the pear. And then on the right-hand side, the top right-hand corner, so that is the starch granule, okay? And then for the one at the bottom left-hand corner, so that is the crystal from the pineapple. And then for the one at the bottom right-hand corner, so that is the crystal from some citrate, the beets or this spinach, okay? So actually what you can be able to see is from those kind of residue, from those kind of characteristic cell type, you could be able to tell, okay, what kinds of food has been taken by the victim. So here, that is a situation that after 2 hours, within 2 hours, you still could be able to see, okay, the undigested food. Okay, so it will be quite straightforward that you can be able to tell what kinds of stuff have been taken. But after 2 hours, so all the food has been digested and then go through the intestine already. But still, okay, you could be making use of those kinds of characteristic cell type, okay, that is the residue inside the stomach, to be able to tell, okay, so what kinds of plants or what kinds of vegetable has been taken, okay, by the victim. So therefore, it will be a very good information if we are really taking with the stomach, okay, because it will be helping us to see any undigested stuff or any specific cell type to know what has been subjected to the victim. So it is a very good piece of information. So apart from the stomach, actually the spleen, okay, it is another kind of organ that we would like to check. Because for the spleen, okay, it is the origin, okay, it is the organ, okay, that we can take the blood from the dead body. So at the very early beginning, we try to take the blood sample, but that will be available for the alive person. So how about for a dead person? For a dead person, if we still want to take some blood sample, then that will be the spleen. Because the spleen, it is the organ as the blood reservoir. Okay, so our blood, okay, will be mainly stored, okay, in the spleen. So therefore, since, okay, with the blood that appear with the poison and the metabolites, if they are being stored up, okay, in the spleen, actually we can be able to check with whether it contains any poison or the respective metabolites or not. So therefore, for the blood sample, if it is an alive person, then you can take the blood sample. If now it is becoming a dead person, okay, the organ that we can be taken in order to review the blood, that is the spleen. So another kind of sample that you could be checked, okay, that will be the gravity fluid of our teeth, okay. So that will be a similar kinds of logic like our hair and our fingernail, okay. Our teeth also contain a lot of blood vessel at the bottom. So in order to supply with the nutrient, okay, to the teeth. So therefore, for the poison and the organ appear in the blood sample, they may be deposit or appear inside the teeth. Okay, so therefore, you could be seeing those kind of poison or the metabolites there. But actually, for some kinds of poison, especially for the metallic poison, so if they are really deposit, okay, so inside the gravity fluid of teeth, actually you will be seeing that the teeth, the respective appearance, you can see some darkened line. Okay, so that will be something appear, okay, so on the teeth that you could be able to make use of these kinds of indication, okay, to tell it may be, okay, a metallic poisoning. So somehow it will be a certain kind of background information as well. Okay. So from the appearance of the teeth, it will be a background information. If you really take out the teeth, so you check with the cavity fluid, then you will be able to draw out some information from there. So the other important organ, so that will be the liver. Okay, so as far as we all already mentioned, liver, that is the organ for the detoxification. When we are having some poison inside our body, so that will be transferred to the liver, okay, so to the detoxification. So therefore, you will be able to find those kinds of poisoning stuff, okay, so there. So for the kidney and bladder, that is the organ that for the collection of urine. Okay, so actually, if now it is a dead person, you cannot ask him or her to go to the washroom, then how you can take the urine? So that will be the organ for the kidney and bladder. Okay, so therefore there will be something like the spleen, okay, for the blood sample. For the urine, that will be the kidney and the bladder. So for some of the poison, as far as we mentioned, we have the gas poison. We also have the volatile organic poison. So mainly, okay, it is not coming through orally or not coming through the injection. You try to breathe in, okay, then the organ to be deposited, that will be our lung, okay. So therefore, for the lung sample, if you are suspected with the gas poisoning or the volatile organic poison, the nerve gas, then taking the lung sample will be quite worthy. At the same time, if you try to take the lung, okay, then actually you can think about the brain as well. Because when we try to breathe in something, if they are quite small enough, they will be go through the blood-brain barrier, then go to the brain directly. So therefore, for the gas and also for the volatile organic, so that will be the lung and also the brain, then that will be a suitable organ, okay, so for the respective checking. For the last one, that will be the fluid, okay, so from our eyes. So there will be also a quite similar things with our teeth. Because from our eyes, okay, so at the eyeball at the back, we have a lot of blood vessel, okay, to supply with the nutrient. Actually, if now there are certain kind of poison or metabolites, they will be just accumulate, deposit, okay, inside our eyes, okay. So therefore, for the eyes, okay, you could be able to find those poison or the metabolites. And for the eyes, okay, it will give you certain kinds of uniqueness that that will be our last kinds of organ to be decomposed, okay? So mainly if a person has been died for a long time, okay, so you may be able to see the skeleton, the bones only. But before it really turns into a bones, okay, actually the last kinds of organ to be decomposed, that is your eyeball, okay? Because for the eyeball, okay, actually it's being protected by your eye socket and also for your scalp, okay? So therefore, it will be really protected that the microorganism will not be able to attack your eyeball quite readily. But for your whole body, that will be quite ready. So therefore, for those kind of decomposed body, the eyeball, that is somehow our last tools, okay, to check with the poisoning cases. So if the eyeball has been decomposed, then actually only skeleton will be remain. So from the skeleton, from the bone, you may try to check with the bones, okay? But for the bone, the information that we can obtain will be quite tiny, okay? So actually with the bones, actually we can be able to tell nothing. So therefore, for those cases have been really died for a long time, there is no way for us to do anything. Okay, so that is the information from the organ. So now, okay, just go through a little bit, okay? So if now, okay, it is a dead person, but we cannot be able to take the organ, do we have anything that is extra or to be supplementary? So the idea that will fall into the forensic entomology. For the forensic entomology, entomology meaning that is related to the study of insect. So by the use of the insect, that will also be used, okay, for the toxicology, okay, because mainly that if now somebody has been killed, okay, he or she is died already. So basically some insect will get to the dead body and then lay out the respective eggs. The eggs will become the baby insect. The baby insect will somehow intake the nutrient from the dead body and then it will grow to become the pupa, and then it will become the adult insect. So actually, those kind of insect will be just somehow live along, okay, with the dead body. They will inject, okay, the dead body and then to be grown up. So therefore, right now you could be imagine if now, okay, a person has been poisoned and then die right here. The insect comes, okay, then the baby insect will eat the dead body. So if now poison appear in the dead body, basically the insect will also take up the poison. So therefore, right now, what you could be imagine quite directly is the insect itself, okay, would bear some of the poison that is coming from the dead body. So if we could be able to take the insect back to the lab to conduct the poison analysis, if we could find some poison there, so basically we can be extrapolate to say, okay, the dead person has been poisoned, okay, because the nutrient, okay, from in the insect should always be coming from the dead person. If poison are there, then meaning that the dead body contains the poison. So not only checking with the insect itself, okay, we can be checking with the growth cycle of the insect. For example, like for this kind of blowflies, okay, so in the life cycle, so when the person is die, okay, then for day one, the blowfly, okay, will lay out the egg on the dead body, and then day two, it will become the baby, and then at around 1 week later, then they will become the pupa, okay, and then 2 weeks later, then they will become the adult insect. So that will be a well-known life cycle, okay, for those kinds of insects. However, if now the insect has been poisoned, okay, so somehow by the dead body, okay, actually the growth cycle will also be changed, okay, because if someone is being poisoned, the life cycle should has been changed, okay, it will be quite straightforward logic. For example, like after 2 weeks time, okay, you cannot see the adult insect, so they are still baby, okay, the whole growth cycle has been altered, has been changed. That will be another kind of indication for the presence of the poison, okay, because now the insect has been poisoned, then it cannot grow anyway, so therefore the growth cycle will be changed a lot. So with the use of the insect, with the use of the entomology, basically the same as the organ issue. We cannot tell any short-term or long-term poisoning because what we can say is there are some kinds of poison from the dead body only. And then for the relevancy, that will be identification only, okay? Because now, okay, there will be a lot of factor from there. So the dead body contains some poison, that will be the accumulation or the deposition rate of the poison as well as the original victims, the excretion rate. For the insect itself, it will take in the poison. At the same time, they will excrete out the poison. So therefore, there are a lot of individual parameters there that the concentration will be meaningless. So for the poison and the metabolites will be both be appear there. So no matter what kinds of things in the body, they will be appear in the insect. So therefore, they will be somehow the same thing. And for the availability, that will be available for a dead person. Okay, so only with a dead person, the insect will come to lay out the eggs. So in order to collect the insect, okay, so that will be quite basic to do so. So for the insect, there are 3 different kinds of form: the baby, and then we have the pupa, okay, and then we have the adult, okay. So for the baby and the adult, okay, so for the adult, they can fly. Then you are going to make use of the hand net, okay, in order to collect it. And then for the baby, okay, just try to make use of a forceps, and then you could be able to take it. So with the adult and the baby, so basically you try to put it into the ethanol solution as the preservation. For the pupa, okay, so you are going to put it into an open virtual container because they are somehow, they are quite stable, okay, they are just staying as itself somehow for some days. So therefore, no need to do any preservation, okay, so just take in an open virtual container, and then you take back to the lab to deal with the further analysis. So after you collect with the adult, collect with the baby, collect with the pupa, after you remove the dead body, for the last tools that you need, that is to make use of the soil sample, okay? So soil sampler to grab some soil underlying the dead body. So basically, for that person, okay, has been died for some time, okay, he or she will be decomposed. So you may have certain kinds of decomposition fluid, okay, so that is being leached out. So those kinds of decomposition fluid will be just getting into the soil, so that you try to collect the soil sample. So why, okay, we try to collect those kinds of decomposition fluid? Mainly that the body contain poison and metabolites, so that those kind of decomposition fluid will also contain the poison and the metabolites, okay? So therefore, it is another kind of sample that you would like to take, okay, in order to check with the poison analysis. Okay, so maybe I let you have a 10 minutes break first, okay, before we go forward to talk about the protocol for the poison analysis. Okay, so let's come back after the break. Okay, so can you still hear from me? Okay, so thanks for the reply. Okay, then I assume everything is still working. So for the last 1 hour, actually we have been finished to talk about the biological sample that we need, okay, for the poison analysis. So basically on the whiteboard, so you can see we have six, okay, so different kinds of sample that we can use. So from the alive person, we can make use of the blood sample, urine sample, hair sample, or the saliva. So for the dead person, then we can take out the organs, or even we can sample the insects, okay, so for the analysis. For the organ, we have been mentioning some important organ, for example, like the stomach or the spleen, okay, for the blood sample, the kidney and bladder for the urine sample, the liver, okay, that is the organ for the detoxification, or even for the gas or the volatile organic poison, we try to take the lung or the brain, okay? So for the last kinds of organ that we can take for a decomposed body, that could be our eyeball, okay? So we have been finished to talk about those kind of sample. So during the break, okay, we have been encountered one kind of question, okay? So basically for the blood sample, we can be making use of a syringe to draw out the blood and then we put it into the renal jet tube. For the urine, just provided with the plastic container, ask the guy, okay, to go to the washroom readily. For the oral fluid, it could be the cotton blood, okay, to scrub the saliva, or even open the mouth wide to let the saliva to be dropping out to the plastic container. For the hair sample, now, okay, the question that we encounter is, after we cut the hair, how, okay, we contain it, okay, because the time zero, that is really important. If we just get it into an opposite, okay, let's say that is the original 0 point, but we mistaken that is the 0, then actually the whole time frame, okay, will be in an opposite manner. So therefore, normally, okay, so for those kind of hair, we are going to have some paper, okay, so to contain it. And then it will be telling you that, okay, so for this end, okay, that will be the end, okay, so of the hair, okay. So therefore, what you try to do is imagine you have the hair sample, so that will be the follicle, and then you will be just put it like this way, okay. So therefore, we'll be indicating to you that that is the end, okay, that is the beginning. So therefore, we have this kind of specifically made, okay, paper, okay, in order to contain the hair sample. So after you put the hair sample, okay, onto the paper, you just try to wrap it, okay. So you will see, okay, the end and the beginning because there is a direction there. So I hope, okay, I answer the question from the student, okay, so in the chat room, okay, to letting you know, okay, so how we contain the hair sample being cutting out, okay, so from the victim or from the suspect. Okay, so any further question at this moment before we start to talk about the analysis? Okay, so for the analysis right now, that is the poison analysis. So if you are checking with the general scheme, okay, that is quite simple, okay, or quite similar to the one that we have been talking about in the drug analysis. So the first thing that we need, okay, so somehow that will be the screening test to narrow down the number of possibility. So secondly, we have to go through the sample pre-treatment, okay, in order to obtain the sample solution. And then we have to go through the confirmatory test in order to identify what it is, at the same time quantify how much are there. So at last, okay, we have to run through the retest, okay, in order to achieve the double confirmation. Actually, the general scheme that is quite similar. And the technique, okay, that we use, okay, some of them, they are shared within the two topics. For example, like in the screening test. So we could be able to make use of the color test or even the microcrystalline test in order to screen out the drug poisoning cases. Because for some of the poisoning cases that is related to the drug, okay, especially that will be the abused drug poisoning cases. Okay, so some people, they are abusing a drug, they are addicted to it somehow with a very strong dependency. So they may be having the overdose so that they will be being poisoned. So therefore, there will be a drug poisoning cases. So we could be just applying the color test, okay, the Mayer's reagent as the preliminary color test. If it is positive, then we go through the Marcus reagent or the Scott reagent for the opium derivative and vitamins, ecstasy, or the cocaine. And for the Mayer's reagent, if they are negative, then we go to another side. So let's say we could be trying the Daly Copenhagen reagent for the barbiturates, or even for the enriched reagent for the LSD. So with the plant material, we can be trying the quinolidine reagent for the THC. So that is the marijuana. So if you have a lot of sample left behind, okay, even that you could be trying the secondary color test, for example, like the Markey reagent to double confirm that is the opium derivative, or even with the KN reagent, then you can be checking the THC as well, okay? Or even now we have the Simon's reagent so that you can tell apart whether that is the amphetamines versus the methyl amphetamines. So actually you can be applying the color test in this way, okay? So as the drug poisoning cases for the screening. For the microcrystalline test, that will be still the same. Maybe you are adding in with the gold chloride, gold bromide, okay, in order to build up the crystal, then you observe it under the dark field microscope in order to tell what kinds of drugs are there. But by using the color test or the microcrystalline test, that will be only subjected to the drug poisoning. So therefore, the scope will be quite narrow. In order to make it somehow widely applied and to make it more universal, so we may try to make use of the biosensor, especially that will be the use of the enzyme or the antibody, so that we can be checking the poison somehow in a universal way. So still keep that in mind, okay, in the poisoning cases, that is not only the drug poisoning or the organic compound, okay, so for the poisoning cases, we have the metallic poison. So actually we have a kind of screening test that is the range test, okay, in order to screen out whether that is a metallic poisoning or not. So therefore for the next around a few slides, I will talk about the biosensor and the range test, okay, somehow in details. For the color test and the microcrystalline test, you can be checked back from the drug analysis. So after the screening test, okay, we are going to go through a very rigorous extraction. So bear in mind that right now we are dealing with the biological sample. What we expect is the matrix effect that is quite severe. So therefore, we have to completely isolate our analyte, okay, the poison, okay, that we analyze away from the matrix. Otherwise, with a severe matrix effect, nothing that we can do. So therefore, I will tell you how we can be playing with a very rigorous extraction. So using the liquid-liquid extraction and the solid phase extraction. For the confirmatory test, basically for the chromatography, spectroscopy, and the mass spec, they are the same kinds of things, okay, that we have been talking about in the drug analysis. So just one thing more is, right now in the chromatography, okay, we are going to make use of the HPLC as well, okay, because we now have something that is non-volatile, something that could be thermally unstable. So therefore, for the chromatography, okay, it doesn't to be the GC only, it could be the HPLC as well, okay? But for the chromatography, for the spectroscopy, for the mass spec, I guess, okay, you can be learning back from the drug analysis. So that in the following slide, I will mainly focus on the atomic spectrometry. Because right now we have the metallic poison and some of the poison, okay, metallic one that is radioactive. For example, like I mentioned to you that with the plutonium-200 or even the nickel-63, so that is really, we need to apply, okay, certain kinds of isotopic analysis, okay, so in order to tell, okay, what kinds of metallic poison are there. At the same time, I will show you some more details with the speciation, because especially for the metallic poison, for the arsenic, we have the arsenic 3 oxide, arsenic 5 oxide. For the mercury, we have the metallic mercury versus the organic mercury. Actually, their toxicity, they are quite different, okay? So therefore, we have to know about the speciation as well. So at last, okay, I will show you two miscellaneous cases, okay? So that will be one on the carbon monoxide poisoning, the other will be on the drunk driving, okay? So to start with, let us first talk about the screening test, okay, by the use of the biosensor. So for the biosensor, basically the most important component in the biosensor, that will be the bioreceptor. So the bioreceptor will be quite selective and quite specific toward a certain kind of poison. So imagine that now we have two pens, okay, on my hand. So we have the blue one, we have the green one. So you could imagine my hand, it is a bioreceptor, okay? So it will sense, okay, the blue pen only. So when the two colors come over, the bioreceptor will be grabbed, okay, the one that is being targeted. For the other, okay, it will not grab, okay? So once, okay, the bioreceptor grab the target analyte, that is our poison, then when they combine, signal will be produced. Okay, so that is somehow the basic principle for the biosensor. So right now, if we have a lot of different kinds of compound passing through, if they are not matched, actually it will not grab anything. So therefore, no signal will be given. Okay, so you can see black, green, and red. Okay, they are not being targeted. So no signal will be given. Once, okay, with the presence of the poison that is target, so that is the blue color. So the bioreceptor will grab the blue one, the other will be just free. And then with the combine of the bioreceptor and the target poison, then signal will be generated. So that is somehow how the biosensor works. Those kind of bioreceptor could be a certain kinds of enzyme, could be a certain kinds of antibody, it could be a DNA, it could be a certain kinds of bacteria, cell, or even tissue. But for our interest, okay, we try to focus on the enzyme and the antibody only, because they are quite sensitive, okay, to the poison. Okay, so enzyme basically it can be sensing to the poison, that the poison will be making the enzyme activity become lower. So it is somehow one way to target. For the antibody, they will sense, okay, with the antigen. Okay, you can be imagine that the poison itself, it is a certain kinds of antigen that make our body to be harmful or to be something that is no good. Okay, so therefore it is a certain kinds of antigen there, and then the antibody will sense the antigen that is the poison. So that is the reason why the enzyme and the antibody, that is the common kinds of bioreceptor that we use, okay, for the screening test as the forensic toxicology for the poison analysis. So let's, okay, go through some details, okay, with the bioreceptor making use of the enzyme. So by making use of the enzyme, the assay that we are using, that is the enzyme inhibition assay, okay. So to make it short, okay, so it will be just saying to be the enzyme inhibition assay, okay, so that is the EIA. So enzyme inhibition assay. So for the assay right here, basically we have the poison there. So with the targeting enzyme, okay, so the signal, okay, of the sensor will be decreased, okay. So mainly that if you are now checking with the PowerPoint slide, okay, so inside the sensor, we have a certain kinds of enzyme, okay, and some kind of substrate has been provided. So with the enzyme working normally, okay, it will be just reacted with the substrate and produce a lot of products, okay. So those kinds of products you can be regarded as a certain kind of signal. So with the presence of the poison will just occupy the space on the enzyme, or even you will say that the poison now poison the enzyme, okay, making the enzymatic activity to become lower, so that less products will be generated with the substrate being provided. So therefore, with a decrease of the signal, you can be able to tell, okay, whether the sample contain any poison or not. So if you try to make it like a time curve, Okay, so basically we have the y-axis versus the x-axis. So that is the signal, and then that will be somehow like the time. Okay, so at the very early beginning, so you are seeing a lot of signal. Okay, so nothing there. Then the sensor just working normally. Okay, substrate combined with enzyme and then a lot of signal. So once, okay, the sample has been added with the presence of the poison, then the signal will be decreased. Okay, so will be decreased. So with seeing the decrease of the signal, okay, then you could be able to tell, okay, there is a presence of the poison. But at the same time, the magnitude of the decrease of the signal, okay, that will be somehow proportional to the poison concentration, okay? Because what you can imagine is if you have a lot of poison, then the enzymatic activity will be even lower. If you just have a tiny amount of poison, then the enzymatic activity will be just lower a bit. So therefore, the decrease of the signal will also be proportional to the poison concentration. So that will be a very good pieces of information that at the screening test, not only narrowing down the number of possibility, it also gives you a tentative amount, okay? Because how sensitive that we need, okay? Actually, at the very early beginning, we don't know. But with now, okay, the preliminary concentration, then we now know, okay, whether that is at the ppb level or it is in the ppt level. So therefore, it will be a better information for us to select an appropriate instrumentation for the confirmatory test. So that is the enzyme inhibition assay. So basically, poison together with the enzyme, that signal will be decreased. So the other form, we are now making use of the antibody, okay, so as the bioreceptor. So that is the ELISA, okay, so enzyme linked immunosorbent assay, okay, so that is the ELISA. So for the ELISA, now we have the poison, okay, so with the poison and the bioreceptor that we are using, that is a antibody. So once, okay, the antibody sense the targeting poison, then they will combine, okay? So I'm not trying to bother with you for those kinds of details, but when they combine, okay, so basically color will be given, okay? So you will be seeing the color, okay? So you will see a color change. With the color change, then there will be a positive result to be tell, okay? So there are certain kinds of poison. But right now, for the ELISA, the color intensity, okay, that is the absorbance. So that will also be proportional to the poison concentration as well, okay? Because what you could be imagine is, if you have a lot of poison, the color will be some more intense. If you have less poison, the color will be less intense. So therefore, based on the color, you still could be able to achieve the same thing. Not only identifying that we have something that is a screening test, at the same time, that will be proportional to the concentration that will be a preliminary concentration. So that is somehow a reason why biosensor that will be somehow much better, okay, than we just try to apply the color test or the microcrystalline test, okay, as the screening for the poison analysis. Because right now it is really a poison, no matter that it's a drug poison or it's like whatever kind of poison, it will be somehow much more universal that depending on what kinds of bioreceptor that we are using. Okay, we could be using some specific enzyme, we could be making use of some specific antibody, but it will be universal to that. The other pieces of information that will be the preliminary concentration. Okay, so this is something that cannot be given from the color test or the microcrystalline test. So with the enzyme inhibition assay, with the ELISA, they are something good. Okay, so actually it is quite easy to do. But by using the enzyme inhibition assay and also the ELISA, actually you cannot perform the simultaneous analysis. What it means is, for one kind of enzyme, for one kind of antibody, actually they will be sensing one targeting poison. So with the assay right here, one assay, one poison, okay, but right now, for the poison analysis, what you could be imagine is we have a lot of different kinds of poison. Now this is the screening. If trial 1 it is negative, then we have to try the trial 2, try and trial 3. Actually, there will be try and error basis. Okay, so in order to making the throughput to be higher, actually we need to think out a way that could we achieve the simultaneous analysis during the screening. So that is the reason why people invent, okay, the strip of latent flow immunochromatographic assay. Okay, actually that is a test paper that you could be embedded or stabilize certain kinds of bioreceptor on some position. Okay, so on the paper. So therefore, one antibody or one bioreceptor at one position and other bioreceptor at the other position, so that with the sample added, they will be just going through the test paper. So they will be run on the test style number 1, test style number 2. So different position will be different kind of poison, so that you could be having the simultaneous analysis. So that is really important, okay, to be observed. Okay. So actually the mechanism that is the same, okay, as what we have been using recently. So that is the RAT for the COVID. Actually, we try to making use of the poison, okay, so sensing to an antibody. So if they are combined, then they will be stable on the test line. If they don't, then they will go to the control line. Okay, actually, more or less, okay, that is the same kind of principle, but we just try to do a modification that now the antibody that we are using, okay, in the immunochromatographic assay that is sensing to the poison. So just try to show you with the video, okay, to let you know what happened. So you can imagine, okay, the virus particle, it is a certain kind of poison that you will know, okay, what happened to it. Okay, so from the video, I guess you know what happened. Okay, so imagine the virus particle, it is a kind of poison. Okay, so it will bind to the original antibody. So if we have the poison and the antibody together, that will bind to the test line, so that you will see the light there. For the rest, okay, they will be binding to the control line to be telling you that the result that is valid. Okay, so if later on you don't have any poison, so actually those kinds of antibody, they will be just binding to the control line only, so nothing will be appear on the test line. Okay, so actually for the immunochromatographic assay, so that is a kind of test that we can be able to achieve the simultaneous measurement. Okay, just depending on how much test line are you having. For each of the test line, there could be a certain kind of specific antibody to targeting with a specific poison. Then actually we're making your life, okay, to be much more easier, because by the original setting, okay, the enzyme inhibition assay and the ELISA just one at a time. But right now, for the immunochromatographic assay, it can be 2 at a time or even 3 at a time, just depending on how much, okay, test line are you having, okay, on the strip. So the other kinds of screening tests, okay, that we can use, that is the range test. Okay, so I'm trying to make use of another color, because for the range test that will be applied to the metallic poison only. Okay, because right now, please be bear in mind that for the poison, it could be now, okay, in a metal. So for the range test, basically the poison, okay, so now we are having, so that is metallic in nature, okay, and the reagent that we use, okay, will be a copper metal, okay. So the metallic poison we added to a copper metal, if it is really a positive result, we will be able to see some deposition. Okay, so some deposition of the metallic poison on the copper metal. So that is somehow how the range test work. Okay, so normally, okay, the metallic poison that we are concerning could be the antimony, arsenic, bismuth, selenium, thallium, and also the mercury. So basically, we try to dissolve, okay, the sample into a concentrated hydrochloric acid, and then we try to add in the copper metal. So actually, that is somehow like a replacement reaction that you learn from high school. Okay, if now we are using the copper as the somehow like electrode or copper as the surface. So if now we have some dissolved metal, okay, in the solution, if they are somehow affinity to the copper, then they will deposit there. Okay. So therefore, for the appear of the antimony, arsenic, bismuth, and the selenium and thallium, if they are deposited onto the copper metal, then you will be seeing the dark coating. For the mercury, that will be silver, because mercury by itself it is silvery. Okay. So actually on the screen, okay, at the right hand side, you will be seeing those kinds of appearance. So from number one, that is the copper metal. With the presence of the bismuth, arsenic, and the antimony, you will see, okay, the copper, the surface has been darkened. Because the bismuth has been deposited onto the copper, arsenic has been deposited onto the copper, or even the antimony has deposited onto the copper. It will be the same thing for the mercury. Mercury deposit on the surface of the copper, but in real mercury, that is a shiny metal, okay, so they will be appear with a silver coating. So therefore, for the range test, okay, it will also be used for the screening purpose, but it will be in a way that if you suspect that it is a metallic poisoning cases, then you are making use of the range test, okay, for the checking. Okay, so that is somehow the things that I want to share with for the screening test. So after the screening test, basically, we are now tentatively knowing what are there. But before we really subjected to the confirmatory test for the chemical instrumentation, so we have to prepare our sample solution. So to prepare with the sample solution, we cannot just simply make a complete dissolution, try to make use of appropriate solvent to dilute it, and then we try to measure it. Actually, there is no way to do it, okay? Because if you just dilute it, concentration getting even lower. At the same time, after the dilution, the matrix still there, and then the matrix effect that is severe, and then actually the result will be quite sour. So therefore, for the sample pretreatment right here, that will be quite rigorous to achieve, okay, a sense that we have to completely isolate our analytes away from the matrix. At the same time, we need a very good recovery. If after the extraction, something that is not being recovered, actually you are losing your sensitivity. So therefore, there will be two main goals, okay, so in the sample pretreatment. So extract out the analyte completely isolated away from matrix, and also completely that is for the recovery of 100%. So imagine that most of our sample that we are dealing with the poison analysis, they are of a liquid sample. The blood sample, urine sample, saliva, actually they are of a liquid sample. So therefore, to be dealing with the liquid sample, only commonly two different kinds of extraction can be used. That will be the liquid extraction and also the solid phase extraction. For the other, okay, you may learn from Dr. Who, okay, we may have the supercritical fluid extraction, SORT-SET, or even the SORT-TET extraction. So those kind of stuff, they are applied to the solid sample. So right now, we are on the liquid. Okay, so therefore, only liquid extraction and the solid phase extraction could be applied. So simply, if you are saying with the liquid extraction, so that is the LLE. So for the liquid extraction, quite simple that we are having the sample solution. So inside the sample, we have the analyte. Okay, so for the analyte, just try to assume that that is polar in nature. And for the matrix, we have the polar matrix and also the non-polar matrix. Okay, so that is somehow what happened, okay, in our sample solution. So for a typical usage of the LLE, so basically, if now the analyte that is polar in nature, then we try to add in, okay, the polar extraction solvent. So we try to add in a polar solvent in order to take out the analyte. So if we just do it in this kind of simple way, analyte polar will be taken out, and then the matrix also polar in nature will be taken out. Only the matrix that is non-polar in nature will be stay behind. So therefore, right here, what you could be imagine is you are now able to separate some kinds of matrix away, but it didn't be complete. Because from the extraction solvent, you still can be seeing the analyte will be together with the extractable matrix. Okay, so therefore, what we have been assumed is for those kinds of biological sample, the matrix effect that is really severe, then with the presence of the matrix, it doesn't help. So therefore, by using this kind of approach, okay, so just based on the polarity, okay, so of the analyte, actually, we cannot be able to completely separate our analytes away from the matrix. So therefore, for the LLE extraction, we need to think up of another way to do it. So on the slide, actually, you could be seeing that apart from the polarity, we will also trying to make use of the nature of the analyte, that is the acidity and the basicity, okay, to help us to get rid of the matrix. So what are they? Basically, for some of our analyte, they may be paired with the carboxylic acid functional group. Actually, under certain kind of pH environment, they will become COO minus. And for some of our analyte, that will be a amine group, okay? So under certain kinds of pH, that will become NH3 plus, okay? So actually, if you learn about the liquid-liquid extraction, if the species that is bearing a charge, actually, if it is matching with the polarity, it won't be being extracted, okay? Because of the charge separation, they will always be stay in the sample, okay, instead of getting to the extraction solvent. So therefore, with the nature, okay, by adjusting the pH, that will be help us in order to facilitate the complete isolation. So what it means is, at the very early beginning, we try to change the pH of our sample, okay, according to the acidity or the basicity of our analyte. So let's say, okay, if now the analyte pair with the amine group, we try to lower down the pH so that our analyte, okay, so now it become positive, okay? And for our matrix, there will still be the matrix, so nothing changed there, okay? So at this situation, after lower down the pH, your analyte now bearing the charge, okay? So the next step is we try to add in the polar extraction solvent, okay? So now only the extractable matrix, the polar matrix will be extracted, because for those kinds of things matching with the polarity, but it has a charge, it will be just staying in the sample. So therefore, only extractable matrix will be being taken out, okay, for this time. So after you remove, okay, the first layer of extraction solvent, then you try to readjust the pH back to normal. Then the analyte now become neutral and now bear with the non-polar matrix. So further on, you try to add in the organic solvent again, okay? So therefore, only the polar analyte, our targeting poison, will be extracted. Because for the non-polar one that is mismatched, then it won't be taken out. So therefore, right here, what you can be see is with those kind of adjusting pH, we could be able to isolate our analytes away from the matrix. Because at the very early beginning, we are quite concerned with the extractable matrix. But right now, we are making use of a pH, Okay, in order to make the extractable matrix will be extracted at the first time, then our analyte will be staying behind. We apply the LLE in the second time so that only analyte will be taken. So therefore, with this kind of approach, together with the polarity and also the nature, okay, so the extraction will be complete in a sense, okay. Now, no more matrix will be there in the sample solution, then everything will be perfect. So other than the polarity together with nature, if you still recall a little bit from the sample P treatment, for the LLE, we do have the single extraction. So you do the extraction once only. And also we have the multiple extraction so that you try to divide the extraction solvent into several equal portions. And then you perform the extraction for several cycles. So actually, with the use of the multiple extraction, the recovery will be approaching 100, okay? So therefore, multiple extraction somehow would be preferred, okay? So that we aim for a high recovery rate, okay? So that is the approach, okay, how we can deal with the LLE. Actually, you can see the whole approach will be quite comprehensive and tedious, okay? So polarity together with the nature. So not only adding in the extraction solvent, you have to adjust the pH as well. And to play safe, multiple extraction will be applied, okay, in order to maximize our recovery. So for the SPE, okay, so that will be the solid phase extraction, okay. So for the solid phase extraction, that will be an exhaustive extraction method so that we are not quite worried about the recovery, but mainly we still need to be dealing with the extractable matrix. So originally, you can be imagined in the samples, in our sample, we have the poison that is polar in nature. At the same time, we have the matrix also polar and also matrix that is non-polar. So if now, okay, this is getting into the solid phase cartridge. So let's say the cartridge that is polar in nature, okay? So after we load in our sample solution, so the polar analyte and also the polar matrix, okay, will be checked, okay? For the non-polar matrix, they will be just getting away. So normally, by using of the SPE, you are going to elute, okay, with a very strong affinity solvent. What it means is, if now you are eluted out with the strong affinity solvent, so all the things trapped inside the cartridge will be eluted out at the same time. So you will be having the polar analyte and the polar matrix together. If you just, okay, elute it once only. So it is not perfect, okay, because it is somehow like the situation for the LLE with the polarity only, okay. So therefore, how we can be making use of the SPE so that we can be isolate our analyte away from the extractable matrix. The trick, okay, that we are using, okay, that is the step-by-step elution. Okay, so step-by-step elution. Instead of using a very strong solvent, we try to differentiate out, okay, so the polarity, okay, of our analyte. So let's say for the matrix, okay, that is somehow weakly polar, okay, and for our analyte, that is strongly polar. So the polarity, although they are the same, but they still have the degree of difference, okay, so strongly polar and weakly polar. So therefore, now what we are doing is we try to making use of different kinds of solvent for the elution, okay. So maybe at the early time, okay, we try to make use of a weakly polar solvent, okay, so that only the extractable matrix will be getting away, okay, so that will be weakly polar solvent. So after that, okay, we have our analyte, okay, live away, and then we try to apply a strongly polar solvent, okay, in order to elute it out. So therefore, what you can be see is instead of just using a polar solvent, okay, So for the illusion, so we try to make use of a weak polar solution for the first illusion, and then we have a strong polar illusion solvent, okay, so at the second time. So therefore, there will be a step-by-step illusion that we try to get rid of the extractable matrix. But all in all, okay, no need to worry about the recovery, okay, because all the things will be checked. If you are using a correct, appropriate illusion solvent, everything will be eluted out. But the main idea is you need to make a differentiation of our analyte towards the extractable matrix. Otherwise, okay, if they are eluted out together, then actually the issue didn't be solved. We have to somehow make the illusion to be with the degree of difference, okay? So weakly polar, middle polar, strongly polar, so that they are being eluted at different steps. Then we are going to achieve, okay, with a solution that only contains, okay, the analyte only. So that will be perfect, okay? So now no more matrix, then no need to worry about matrix effect. So from the sample solution, only analyte, then that will be quite ready, okay? So for the confirmatory test for the instrumental analysis. Okay, so we have been finished to talk about the sample pre-treatment, okay? So any questions or problem at this moment before we still keep going forward? Okay, so seems no reply, then I assume no question. So right now we go to the confirmatory test. So as I already mentioned, for the confirmatory test, basically we can make use of the chromatographic technique, the spectroscopic technique, and the mass spec. Okay, but right now what has been left behind, that is the atomic spectrometry. Because right now we are having the metallic poison, so that atomic spectrometry could be applied. Okay, so for the atomic spectrometry, we have 4 different kinds of techniques. So like the flame AA, graphite furnace AA, and also we have the ICP AES, and also we have the ICP MS. Okay, they are the 4 kinds of atomic spectrometry that could be applied, okay, to analyze the metallic poison. So for the flame AA or the graphite furnace AA, we try to measure the atomic absorption. And for the ICP AES, we try to measure the atomic emission. For the ICP mass, we try to measure the mass to charge ratio of an atom. So I guess, okay, you have been learned from me, okay, in the analytical spectroscopy to know about the respective principle or the theory or the background, so that I won't try to bother with you, okay, for the details. So just, okay, go to the things that is important. So for the identification, so if you are using the flame AA or the graphite furnace, actually we can identify the analyte by the absorption maximum. So what kinds of wavelength, okay, does the atom absorb? Okay, so meaning that what kinds of wavelength, okay, does the heavy metal absorb? For the atomic emission spectrometry, so basically there will be the atomic emission. So different atom, different metal, they will emit, okay, differently. So therefore we try to sense, okay, for the emission wavelength. For the mass spec, that will be the mass to charge ratio. Okay, for example, like magnesium will be 24, copper will be 63, and then for silver will be 107, bismuth will be 208, okay, lead will be 208, bismuth will be 209. So actually just depending on the atomic mass, okay, of your analyte. For the quantification, atomic absorption spectrometry, AAS, that will be absorptions, okay, so that will be the absorbance. For the ICP AES, that will be the emission intensity. For the ICP mass, that will be just counting the number of ion, so therefore that will be the ion counts, okay. So therefore, we still, okay, can be achieving the quantification using the atomic spectrometry. But all in all right here, all the technique that we are talking about would be destructive in nature. Because right now we have the atomizer, the flame, graphite furnace or even the ICP, okay, it will be just burning up, okay, so all our sample solution, burn up all our sample. So therefore, they will be destructive in nature. So later on, okay, so once 2 slides later, I will show you another kind of technique, okay, that is the X-ray fluorescence. So that is non-destructive in nature. Otherwise, okay, we don't, okay, have any preferred test, okay, for the first confirmatory test. And for the atomic spectrometry, I guess, okay, everyone in class, okay, you know how we can make a comparison, okay? For example, like we can compare with the detection limit. We can compare with the number of analytes that we measure at a single time. And also we could be able to compare with the price of the instrument. So I guess, okay, I don't need to do further discussion on that because you know, okay, so flame APP, M level, graphite furnace PPB, ICP AES will be PPB, and ICP MS will be TPT, okay? And atomic spectrometry, one analyte at a time. For the ICP technique, that will be simultaneous analysis. And in terms of the price, you know, okay, ICP MS will be the most expensive one. And then for the flame AA will be the most cheapest one. Okay, so actually, for all the technique, you can measure heavy metal. But when, okay, you have to use which one, there will be some guideline, okay, for you to make the judgment. But there is one thing that you need to pay attention is all the technique, okay, right here, only the ICP-MS, okay, can give you the isotopic analysis. Okay, so for the other, okay, it will be a total metal content. But for the ICP-MS, it is still a total metal content, but it will also be able to tell you with the isotope. Because if you remember right now for the metallic poison, we have the radioactive poison. For the radioactive one, you do have the plutonium-210, nickel-63. Actually, it has to be that isotope, okay, to be radioactive. So therefore, if you are not having the nickel, but all the nickel that is 60, actually it's a normal nickel. Okay, so therefore, isotopic analysis is needed to confirm whether that is a radioactive poison. So that will be the responsibility for the ICP-MS. So all in all, okay, for the detection, that will be the total metal content only. But as I already mentioned to you that for the some kinds of metal, let's say for the arsenic, so we have the arsenic 3 oxide versus arsenic 5 oxide, or even we have the mercury, metallic one, versus the organic mercury. Actually, their toxicity, they are not the same. If you just measure total mercury, actually you may be drawing a wrong conclusion. Or maybe if you just try to measure total arsenic, the conclusion may also be wrong. Therefore, in order to make a speciation, normally we are trying to couple it with the LC or the IC to the atomic spectrometry. So to be somehow the most sensitive one will be the LC ICP-MS or even the IC ICP-MS. Making use of those kind of separation technique, okay, in order to separate those species into different time and space, so that we can measure them individually with the atomic spectrometry. For example, like if now you are measuring arsenic species, maybe you are using the IC, okay? Maybe the IC that you are using that is negative charge solver, so negative IC, okay? So the chromatogram that you may achieve, okay, that is the arsenic versus time, okay? So basically you will be seeing 2 peaks, okay? For the first peak, that will be the arsenic 3 oxide, okay? And for the second peak, that will be the arsenic 5 oxide. The reason behind is for the arsenic 3, that will be arsenic 3 plus, and then for the arsenic 5, that will be arsenic 5 plus, okay? Actually, with some more positive charge, they will have a much more interaction with the negative column, so that they will be eluted out later. So therefore, if now, okay, you are coupled with the IC ICP-MS, then you could be able to separate the 2 species into 2 different times. Also for the mercury, you may be making use of the LC ICP-MS. Maybe you are using the non-polar column. Okay? So basically, mercury versus time, okay? And then you will be seeing the metallic mercury, methyl mercury, ethyl mercury. So just depending on the polarity. For a somewhat nonpolar species, then they will be eluted out later. The first one will be the normal mercury. And then for the second one will be the methyl mercury. The third one could be the ethyl mercury and etc. So therefore, just depending on the polarity, okay, then they will be being separate. So therefore, right here, what I want to pinpoint to you is not only atomic spectrometry will be used by itself. For some time, if we really need the speciation, we need a hyphenated technique, LC-ICP-MS or even the IC-ICP-MS. So to end this lecture, just try to show you with the XRF for the last thing. For the XRF, there will be the X-ray fluorescence spectroscopy, okay? So with the X-ray beam, they will be locking out the inner shell electron of an atom so that the outer shell electron will fall back to the inner shell so that with the fluorescence will be given. So since, okay, you know that the atomic configuration, they will be quite specific towards different metal, okay? So those kind of fluorescence, those kinds of fluorescence intensity will be unique, okay, towards the individual kinds of heavy metal. So therefore, based on the emission wavelength, then we can be able to know what kinds of analyte are there. And as it is an emission intensity will be used for the quantification. And as I already mentioned, that will be non-destructive in nature because you just send in the X-ray beam and then the sample will not be being destroyed so that they will be non-destructive in nature. So therefore, it will be a very good try, okay, to be used as the first confirmatory test. And for the retest, then select the other. Okay, so that is somehow what I want to share with you for today. So next Tuesday, okay, we will end up with the two miscellaneous cases. And then I will start to talk about the body fluid analysis. So that will be somehow the second last of the lecture material. So if you do have any questions or problem regarding to today's lecture, so just stay behind and ask. Otherwise, we will be seeing each other next Tuesday. So thanks for attending the class and then have a good evening.